Okay, so for those of you who are in class today, we started our disinfectant lab experiment, and we took samples of the bacteria and stuff growing on the bottom of our shoes, and we are going to culture it, on these plates. That's what these are, and we're going to see it grow. So these are what our plates should look like within the next 2 days. This red one, this is a sheep's blood agar plate, and some bacteria feed off of blood. They have an enzyme that hemolyzes the blood, and so doctors will put a culture on here, and you can come back the next day and see a clear area around the bacteria where it's eaten away the blood. So anyway, that's just something interesting. Alright, so take a guess as to what this purple thing is, knowing that each of these little things, this is a bacterium, this is a bacterium, this is a little colony of bacteria. This purple thing is going to be the point of a safety pin. So, and that's just a kind of comparison so you can see how tiny bacteria cells are. So when we took samples off the bottom of our shoes, we wouldn't necessarily be able to see the bacteria on there. So once we culture it, it's going to replicate, many, many times and form a colony which will be visible to the naked eye. So these are the different cell shapes we can have in bacteria. We can have cocci, bacilli, or spirochetes. Cocci are going to be spherical. If you see the prefix strepto, they are going to be in a strip or a chain. So these, it's chain-like. And then staphylo is going to be in a cluster, like a cluster of grapes. Bacilli, it's going to be rod-shaped. An example is E. coli. These are usually singular. Sometimes you'll find them in a chain, not very often. So then spirochetes, it's going to be spiral. So an example would be the syphilis bacterium, which is a spiral. Alright, so if you remember in the lecture we did over domains and kingdoms, we talked about archaebacteria and eubacteria, and we talked about gram staining and different substances of the cell wall. A really important one is peptidoglycan. If you'll remember, this is kind of a starchy mixture of proteins and sugars. So gram-positive is going to be purple, and gram-negative is going to be pink. So gram-positive cells are going to have a relatively thick layer of peptidoglycan, so that's what that purple stain is going to stick to that causes them to be purple. The gram-negative, it has less peptidoglycan, so the gram stain is not going to stick. So we get it to be pink by adding a counter stain, which is going to color it and stick to other things in the cell, just so we can see the two and compare them. Gram-negative, or the pink bacteria, once it's stained, is often going to be more threatening, really toxic, way more serious, so like bacterial meningitis. That bacteria is going to be a gram-pink, or gram-negative. So external structures. So a bacteria can have a capsule, it can have a flagella, and it can have fimbriae. It does not necessarily have to have these. So a capsule is going to be a sticky layer, allowing them to stick to each other, or to just a surface in general. Flagella is going to be just a protein structure that kind of wags around, flips around to help them move. So they can move towards food or away from toxins. That's pretty much it. Those are the environmental signals we're talking about. So fimbriae is going to be hair-like projections, enabling them to attach to surface. So if you learned the word pili instead in the cell structure unit, that's fine. Fimbriae and pili are the same thing. So an example would be gonorrhea. This is how it's going to attach to the cells to infect them. So on the next slide, I have some pictures. Alright, so you can barely see it, but this is going to be a tonsil cell, like in the back of your throat. And this is the bacterium. And then this green, sticky looking stuff is the capsule. So that's how it's going to stick to that tonsil cell. Down here is a bacterium. This stuff is the fimbriae, and these are the flagella. So some interesting things about prokaryotes. Most replicate within 1 to 3 hours, And the genome is one gigantic circular chromosome. So this chromosome is many, many, many times longer than the bacterial cell. It's just going to be wound up real tight to fit inside. So each replication can mutate many, many times. So this is going to lead to a lot of variation in our prokaryotes. So this is why bacteria can gain resistance to antibiotics and stuff like that. Plasmids, they're small circular DNA molecules. It's just DNA. It's not another organism or anything. It's just part of the bacteria. These are going to replicate independently of that one circular chromosome, the one giant one. So plasmids can carry genes to enhance survival. So they can have resistance to antibiotics. They can carry the genes to create endospores, and endospores are resistant cells. They can withstand harsh conditions like really high temperatures, really low temperatures, absence of nutrients, stuff like that. So the cell is going to form an inner layer around the DNA, its DNA, and the outer part of the cell is just going to disintegrate, and you're left with this endospore. And this endospore can just sit there until it deems that environmental conditions have been restored, and it'll reabsorb some water and become a bacterium again. So here's a picture. So all of this scrolly stuff, this is all one giant chromosome that's been packed inside the cell, and you can see it's coming out of right here. So each of these little circles of DNA, these are going to be the plasmids. And over here on our green bacterium, this dark thing in the middle, that's going to be our endospore. You can see that it's working on making those layers that are going to go around and around and around it to protect. So all the green stuff, that's going to eventually just disintegrate, and you'll be left with that endospore. Alright, so this slide, I don't know why it's cutting off the tops of the slides, but oh well. So this slide is going to talk about, This is going to talk about nutrition, so how the bacteria create, their food and stuff. So we can have up here, you can't see it, but it says autotrophs, and here it says heterotrophs. So autotrophs make their own food, and then heterotrophs eat food that's already been created. So it's like, making dinner or going and buying fast food that someone else has made for you. So photo is going to refer to light, chemo, chemicals, and how we're going to write these, this handy-dandy little formula down here. So we're going to say what the energy source is, then what the carbon source is, and then we're going to add troph, and that's it. So photo, auto, troph. Photo, so it's going to have light energy, and it's going to convert the CO2 to food. Chemoautotroph, it's going to use chemical energy to convert CO2 to food. Heterotrophs, so again, photo, light for energy, and heterotrophs, since they eat food someone else has already made, they're going to use light for energy to turn carbon compounds, to use those as a carbon source. So chemoheterotrophs, they take in organic compounds for both energy and carbon sources. I have a really neat slide next, but it looks cut off a lot of it. So I'll give you what page this is in the book, and it has the energy sources along the top, and then the carbon sources along the side, and then however you match those up, it'll tell you chemoheterotroph, photoheterotroph, photoautotroph, chemoautotroph, and it'll kind of explain what those are. So, bacterial reproduction. So binary fission, this is a really important one, and it doesn't even let you see it. That's so frustrating. Okay, so this is binary fission. So this is going to be asexual reproduction. This is going to be it just replicating its DNA and dividing. So each of its daughter cells, it's going to be exactly the same as the parent cell. So this is what happens in bacterial colonies. You have one little bacterium, and it's going to divide into 2 bacterium, which is going to divide into 4 bacterium, then 8, then 16, on and on and on. So this is how you'll have one bacterium form an entire colony, and that entire colony, each of the bacteria in there are going to be exactly the same, genetically the same. So this doesn't mean that bacteria cannot do sexual reproduction, which is what we're going to talk about next. They have 3 ways to get new genes, to transfer genes, all these different things. So transformation. This is going to be taking up DNA from another source. So when we talked about the DNA experiments, how they found out that DNA is a genetic material, you could see that the non-infected bacteria, as they mixed with the bacteria that was infected, they somehow got the pathogenic genes. So this was through transformation. So this can also happen. Oh, actually, that's the next one. So transformation, it's just the switching of genes, pathogenic genes from one to another. It just picks up pieces of the genes and puts it into its own. So transduction, this is going to be, when a bacteriophage, it sticks its DNA in with the bacteria, so this little piece would be the viral DNA. And sometimes when environmental stress, when it activates that viral DNA, it pulls out more than just the viral DNA and goes and puts this whole thing into another bacterium. So these genes right here that were part of the original bacteria's chromosome are now in a new bacterium. So conjugation, this is actual bacteria sex. This is one bacteria has a sex organ called sex pili, and he attaches to this bacterium, and then they transfer genes. So a bacteria cell can only do conjugation if it has the F factor. I don't even know if you need to know F factor, but that's a component of conjugation. Okay, so now we're going to talk about metabolic relationships and oxygen and how those relate to the bacterial cell. So some bacteria are obligate aerobes. So what does this mean? It means they require free O2. They cannot survive without oxygen. You can have facilitative anaerobes, so they use oxygen if present, but they can use fermentation. So this is without oxygen. Obligate anaerobes, they are poisoned by O2, so they cannot be in the presence of oxygen. So they use fermentation or anaerobic respiration. So respiration, that's creating energy. Just as a refresher. So evolutionary relationships. So we have a universal ancestor, all the way back at the beginning. And we think that archaebacteria, so these are those extremophiles. I'm just going to write extremes. And eukarya, we are eukarya, so organisms with membrane-bound nucleuses and organelles. We think that the archaebacteria and the eukarya probably evolved from the same ancestor, and then eubacteria evolved separately. Alright, so that's it for that lecture. We're going to talk about bacteria's importance and then how bacteria relates to disease on Thursday. And we'll go ahead and finish up the lab that day and start a new worksheet.